Hello students, welcome to EPG Pathshala. I am Dr. Shaukat Mir from Hamdard University, Delhi. Today we are going to talk about natural product analysis and overview under the paper Pharmaceutical and Biological Analysis. After completing this module, we should be able to understand what natural products are, their definition, types, and their role in drug discovery, analysis of natural products, what is the need for their analysis, how the chromatographic methods and spectroscopic methods are used for their analysis. And the last of all, we will be discussing recent advances in this field, which include the hyphenation techniques at the top of them. We will also try to go quickly through the challenges as well as the future prospects of natural product analysis. By definition, natural products include unregulated organisms or natural materials. Next are the unmodified natural materials or compounds regulated by different agencies like FDA. A naturally occurring compound that has been chemically modified, which are called as semi-synthetic derivatives, they also come under the ambit of natural products. And finally, a pure synthetic substance which has been designed on the model or the template of a natural compound as an inspiration, those products also come under this under natural product category. Traditionally, natural products represented by they are divided into two large categories. One that comes from mammalian group, so mammalian organisms, and the other that are obtained from non-mammalian species like bacteria, fungi, plants, marine organisms, and other non-mammalian animal species. The role of natural products in drug discovery needs to be understood. Natural products of medicinal value encompass a diverse range of substances. They have different uses. They can be used as drugs, they can be used as dietary supplements, cosmeceuticals, additives, or even as diagnostic agents. Natural products contribute greatly to the history and the landscape of new molecular entities. Mind you that these new molecular entities are the potential drugs, or in the history they have led to the discovery of those drugs. An assessment of all FDA approved NMEs, new molecular entities, reveals an interesting fact that the natural products and their derivatives, they represent over 1/3 of all NMEs that have got approval. And out of these 1/3 natural substances, nearly 1/2 of those are derived from mammals, and a quarter each are obtained from microbes and plants respectively. This slide provides you a list of natural substances along with their general functions. This is not an exhaustive list. So here we can see at the top of it we have these mavastatin or lovastatin, which belongs to class of statins. They are cholesterol lowering components which are obtained from natural sources. In the same way, we can see the second entry of non-steroidal anti-inflammatory drugs like obtained from salicylic acid, which is obtained from willow. Another important natural products are obtained from opium. So these are known as opioids. The compounds that belong to this class are morphine, thebaine, and codeine. Morphine is gold standard for analgesic drugs, to which all new compounds are compared in their activity to inhibit pain. Next set of compounds that come from natural products are atropine, dopamine, ephedrine, and kallin. These are neurotransmitter modulators which have their role in the treatment of several diseases. The next category is of foliates, dicumarol, vitamin K1, and hirudin. They are useful anti-anemic agents, as well as they take care of the coagulation defects with respect to blood. The last three categories of topoisomerase inhibitors, antimicrobials, and antibiotics, the compounds which make our armament against cancer and microbial infections. So tomoisomerase inhibitors like daunorubicin, tetracycline and camptothecin, while as antimicrobials like erythromycin, rifampicin and oleodromycin, while as antibiotics they include cephalosporin, penicillin, kanamycin and monobactam. So these natural products, they account for about 86% of all therapeutic substances obtained from natural products. This is another list that provides you the potential uses of natural products other than therapeutic agents. Like we have carnosic acid and flavonoids obtained from rosemary, grapes, etc. They are potential antioxidants, while as various coloring and flavoring agents obtained from in the form of anthocyanins or some of those aroma oils like citrus oil, ginger, vanillin, they are obtained from plants shown against their name, so they make good flavoring agents. In the same way, some of those fragrances like jasmine oil, rose oil and lavender are other important products obtained from natural products. Specialty oils and fatty acids like gamma-linoleic acid, biodiesel, which is obtained from Potileca olearacea purslane, so they have their own uses in different fields of cooking or even in automotive or as a propellant. In the same way, we have substances which are used in agriculture like pesticides. Neem is a very good source for azadirachtin, insect plant which gives pyrethrins, and tobacco plant which gives nicotine. So all of these, they are pesticides used in agriculture. And the last of all is beverages which are used. Caffeine is used, wheat is used, or even as a stimulant, tea is used as a beverage, which is an important use of these natural products. This slide provides you a sneak peek into the steps involved in the preparation of natural products. So this is a general outline. On the left hand side you have the inputs that go into these steps, and on the right hand side you get the outputs. So in the first step, which is seed preparation step, the raw material in the form of plant parts or entire plants, organisms, or the products from the plants, they make the feed for this step, and initially they may be dried, or the contaminants can be removed, or the unused parts or the non-drug parts, they can be removed. So there it makes the output. The second step is product recovery, where you want to separate the useful constituents from that of the matrix. So you provide different solvents in the form of liquid, or it can be even in the form of supercritical solvents. So they provide you a medium for product recovery, and the output is in the form of residues or exhausted drugs. The third step is product enrichment. So where you resort to different processes which are specific for different products, and you are into removal of impurities, removal of byproducts, or you can get the first crop of the products. And the last one is purification, where you can use solid phase extraction or different types of solvents, so that you get products which are highly pure or relatively pure. So this is an outline or block diagram of different steps that are used. These are general steps used in the preparation of natural products. So now let's go to the natural product analysis. We understand that chromatography and spectroscopy are the conventional unit operations that are used in the separation, isolation, and identification of natural products. These two methods, these two techniques, that is chromatography and spectroscopy, they are also the mainstay of the analysis of natural products. Chromatography consists of any technique used for the separation of complex mixtures, while as they are separated on the basis of relative affinity of the solute towards the stationary phase. So we understand that in while as in spectroscopy, uh, it involves the use of interaction of electromagnetic spectrum uh, to study the matter or substances. Chromatography means, literally it means color writing. So it was first introduced by Sweat in 1905, and it was actually used for the separation of colored substances from the plants. So now, earlier it was a color separating technique, and it is now it has evolved in a method for separating a mixture into individual components by differential distribution of components between a stationary phase and a mobile phase. It also represents an array of techniques capable of dealing with most complex analytical and purification problems of natural products in complex, which are present most of the time in complex matrices such as plant extracts, biological fluids, foodstuffs, etc. This slide provides you a list of chromatography methods along with their separation mechanism. As you can see at the top of it, solid liquid chromatography with a separation mechanism. Adsorption is the basis of several of those chromatography, like column chromatography. Then we have a very old chromatography called paper chromatography, which is amenable to both adsorption and partition mechanism. Then GLC, which stands for gas liquid chromatography, TLC for thin layer chromatography, and other forms of LC. They have both adsorption and partition as their method of separation, followed by supercritical fluid extraction chromatography. So here we use supercritical solvents. So again, adsorption and partition remains the separation mechanism. So besides this, we have two used chromatographies for which partition is the main separation method, which is LLC, liquid-liquid chromatography, and countercurrent chromatography. So the next one is ion exchange chromatography, which is different from the rest of them in the sense that it involves the exchange of ions, while as capillary electrophoresis involves the movement of charged compounds. So the last one is affinity chromatography. Again, it is unique in its features because it uses biological affinity of proteins for the separation of proteins, peptides, and other protein-based substances. Let's first go to the column chromatography. The right-hand panel, we see a glass column, can be of different sizes. It is filled with adsorbent of different types like silica, alumina, ion exchange resins. They can be used adsorbent or the stationary phase. These can be filled by wet packing method or the dry packing method. And then once your column is ready, the sample can be applied as a mixture at the top of it, and then with the help of different solvent mixtures, the components of this mixture, they get separated over the column and eluted through the base, which is an outlet guarded by a stopper. This slide provides you a list of common solvents along with their miscibility, water solubility indexes, as well as their safety values. So it extends from hexane to water. So this is in the order of their increasing polarity. Hexane, which is non-polar, and then toluene to ethyl acetate, we have moderately polar substances, solvents, and methanol and water, which are polar. So this is particularly useful with respect to select solvent as well as solvent mixture with respect to their water miscibility as well as their polarity index. This diagram shows a visual representation of differences between a normal phase chromatography and a reverse phase chromatography. In normal phase chromatography, in the upper panel, you can see that the stationary phase consists of plain silica or silica which is modified by cyanopropyl or aminopropyl groups. So elution strength for these solvents, it goes from weak to stronger strength with hexane to water as we move from hexane to water. So pull these less polar substances, they are not strongly attached to the silica and that get eluted first, while as more polar substances, they are strongly attached to silica or these types of stationary phases and get eluted last. While as in the lower panel you can see the stationary phase and the pattern of elution strength of mobile phases. Here we see that the silica is modified by modifications or like octadecile or octyl or hexyl end capping of silica. So thereby the octadecyl, the interaction with non-polar compounds from ethyl to octadecyl, C2 to C18 columns, it increases, while as the elution strength of the solvents, it increases from water to hexane. So the reverse, the direction is the elution strength which is completely reverse. So that's why we call it as reverse phase chromatography, which is the basis of new HPLC methods. So this is the diagram of HPLC chromatographic system. So it consists of binary system, which has been shown here. It can also be a quaternary system. Two solvents, they are pumped by two pumps. They are mixed in a mixing vessel. Then they are pumped in the form of a mixture onto a column through a pre-column, and sample at the injection port, sample can be injected, and under the influence of this solvent gets eluted and detected on the basis of different physical parameters like UV absorbance or even refractory index. A thin layer chromatographic system, it is a very old system. It is commonly used in natural product analysis. Various types of stationary phases are silica gel G, S, silica gel G, F254 or calcium phosphate. The preparation of these TLC plates is done by pouring, dipping, spraying or spreading. Then it is followed by activation of TLC plates. Initially they are air dried after spreading, and then they are activated at 110 degrees Celsius for 30 minutes. Then the samples are spotted with the help of capillary, and then they are dipped in a trough or a chamber as shown in the upper panel. Solvent is present at the base of it, and the spots, they get resolved on the basis of relative differential affinities. Students, we have already discussed TLC. So this high performance thin layer chromatography, it is an advancement of this basic technique. So in TLC we have several of those limitations like manual preparation of plates, and then manual spotting, and various other limit of speed by which they can be developed. So all these things have been taken care with the development of HPTLC. A number of advancements have been made to the basic method of HPLC to the basic method of TLC in the form of automation in order to increase the resolution and to avoid the manual these interferences and to allow for accurate quantitative measurement. HPTLC differs from TLC in particle size, pore size, as well as the distribution of particles of the solvent like silica or alumina. HPTLC plates are produced from more uniform silica gel particles of 4.5 micrometers. They to form a very uniform thickness plate over glass slides, which are about 10 to the thickness of the silica gel varies from 100 to 200 micrometers. While as there have been automation with respect to the location, procedure and reporting the RF values. The UV light is the main physical method that is used for visualization and densometric determination of the separated spots, while as the calculation of the RF value has also gone more sophisticated with the use of several softwares in these modern HPTLC systems. Let's now briefly discuss about spectroscopy. So spectroscopy is very important part of the natural product analysis. So this stretch, it shows electromagnetic parts of the electromagnetic spectrum. It starts from these gamma radiations or gamma rays and ends somewhere around radio frequencies or microwave. So when we use different parts of this electromagnetic spectrum to study matter or the substances, we call it as spectroscopy or spectrophotometry. So the absorbance of a compound at a particular wavelength increases with the increasing number of molecules undergoing transitions. So there can be transitions which can take place in visible region or ultraviolet or IR region. Spectrophotometry is an important tool in the analysis of natural products. So absorption spectrophotometry in the form of UV-visible IR or near IR region of the electromagnetic spectrum is the basis of analysis of several natural products. We find many reports of the analysis of natural products where the UV visible IR or near IR regions of the electromagnetic spectrum are used. They actually provide key information about the structure of the natural products. So otherwise these are used for the structural determination, but they are also used for the analysis of these substances. UV is the most sensitive method of all these spectroscopic methods, and since the introduction of derivative spectroscopy, the sensitivity of these UV spectrum, spectroscopy has even gone higher, so that UV spectroscopy is now used for the trace analysis also. In case of IR, the Fourier transform has also changed the landscape of its applications. So it has made it far more sensitive than the classical IR, and it has also made these measurements with IR quite fast. So number of times these IR radiations, they pass through the sample and they have improved the results of IR. So another important thing that has come up nicely is the use of near IR spectroscopy in the analysis of natural products. So it has been successfully applied for the natural product composition determination and the product quality assessment and in the production control in industries. So near IR spectrum, they actually provide you a profile, a fingerprinting pattern, so which can be compared with standard drugs for the quality evaluation also. Mass spectroscopy consists of ionizing chemical compounds to generate charged molecules or molecular fragments and measuring their mass to charge ratios. It is strictly an analytical technique that measures mass to charge ratio of charged particles. It is usually used for determining the masses of molecules and compounds, for the determination of elemental composition, and for elucidating the chemical structures of molecules, such as phytoconstituents and new chemical entities. But it has been used as a detector as well in tandem with several of those chromatography methods. A typical mass instrumentation system consists of an ion source, a mass analyzer, and a detector. NMR spectroscopy is a powerful analytical tool. It uses magnetic properties of certain atomic nuclei to determine the physical and chemical properties of atoms or molecules in which they are present. It relies on the phenomena of nuclear magnetic resonance and provides detailed information about structure, reaction state, reaction dynamics, chemical environment, and metabolomics. When such nuclei are placed in a magnetic field, for example H1 or C13 nuclei, they absorb electromagnetic radiation at a frequency characteristic of that isotope. The resonant frequency, energy of the absorption, and the intensity of the signal are proportional to the strength of the magnetic field. So on the basis of the environment of these nuclei, they provide different spectra which provides information about the compound and molecules. Now let's come to these hyphenated techniques. These can be defined as technique where you combine chromatographic methods to spectral methods. Chromatographic methods represent methods for the separation of chemical compounds, while as spectroscopic methods are any methods that provide information about the identity of the substances. Lower panel you can see this block diagram. So we have separation techniques like GC, LC and CE. When they are clubbed with a detection technique like PDA, IR, mass or NMR, so this makes in hyphenation techniques. Prior to this or after this can be added some purification steps or further detection methods can be added to this. So but the central portion represents hyphenated techniques like HPLC, GCMs and other things. This block diagram represents a schematic outline of GCMs. So here we see that a carrier gas enters through the inlet. At the injector, we inject the sample which is taken by the carrier gas onto a column placed in a thermostatic oven, and there is a long column over which the components of the injected sample get resolved and they get eluted and they are taken to the MS detector, and thereby this GLC acts as a separation medium and mass acts as a detector. Another hyphenated technique shown here is LCMS. So it is a representative of even UPLCMS. So which the LC and the UPLCMS, they only differ in their size of the particles in the column. So we have an HPLC column or a UPLC column onto which we inject sample, and these samples they get eluted under the influence of mixture of solvents from the HPLC pumps. They are initially detected by UV or PDA detector, then they are carried forward to a mass spectrophotometer which acts as a detector. So thereby it acts as a hyphenated technique called LCMS. The mass can be of any QTOF or ESI type. This block diagram represents another hyphenated technique called LCNMR. Here the HPLC or the LC system remains the same. NMR acts as its detector. This is another important hyphenated technique represented in the form of a block diagram. So this capillary electrophoresis mass system, here the capillary system, it connects to jars across which we apply high voltage. So on the basis of their different charge, they get separated through the capillary, and UV-visible and mass, they are used as detector. Students, now let's discuss some of those examples where these hyphenated techniques have been used for the analysis of some of those natural products. So first one is determination of schwarzschild by HPTLC. Here the schwarzschild, which is very bitter compound present in Schwarzschild cherata, so it has been detected in different extracts using HPTLC. The solvent system mobile phase is ethyl acetate methanol water in the ratio of 80:15:5. Scanning has been done at 246. So these dark spots at the center, they represent schwarzschild. So they have been estimated in plant samples present in tracks 7 to 23. Tracks 1 to 6 represent schwarzschild standard. So this is the estimation of schwarzschild by HPLC. So here an HPLC system has been used, which was coupled with UV-visible detector. So reverse phase C18 column was used, mobile phase was methanol water 80:20, flow rate 1 ml per minute, and the scanning was again done at 238 nanometers. So we can easily see in the A, this is peak of schwarzschild in standard, while as B represents the schwarzschild peak, or in the case of an ecosystem laterale. GCMs, as I told you, that it is a very strong technique. It has been used for determining the nutritional potential of several of those oils that are used in food. So here the nutritional potential of olive drupe oil, it was determined by GCMs. So these bigger peaks, they represent major fatty acids like oleic acid, palmitic acid, arachidic acid, and DHA, which is docosahexaenoic acid. So these bigger peaks, they are there, and they present intensity of these peaks represent their content present in the oil. GCA has been also be used for the detection of fixed oils, like in this case Nigella, which is black cumin. The composition of fixed oil from black cumin has been determined by capillary GC, and mass has been used as its detector. So based on GCMS profile, different fatty acids were isolated, like linoleic acid, oleic acid, palmitic acid, and icosapentaenoic acid. So these are very useful polyunsaturated fatty acids which are useful for the cognitive health. Another good example of the analysis of oil by GCMS is tea tree oil. Here the tea tree oil has been evaluated and has been found that Indian tea tree oil, it has terpene 4-ol to the level of about 50%. So it is much above than the standard content recommended by the Australian plant standard, as the content prescribed by the Standard Association of Australia, so which limits it to that it should not be less than 30%. So Indian tea tree oil contains around 50% of this constituent. This is an example where a non-terpenic active compound in images has been isolated and determined on the basis of GCMS. So this study reports the presence of bergaptan as a major non-terpenic component along with icosane, N-icosanol, docasanol. So this constituent is a furanocoumarin and is especially useful for against leukoderma. So its presence in images rightly justifies its use against leukoderma. Now let us discuss the challenges and the scope of natural product analysis. The high throughput screening programs which require more efficient and sensitive methodologies, they are taken up by several of those research institutes to yield adequate information on natural products. So this is one of the scope that high throughput screening is going on in several of those research institutes. Number two is we are lacking in the availability of sensitive and universal detection techniques, which is suitable for both qualitative and quantitative analysis. So the quest or the search for universal and sensitive detectors with uniform response factors for all analytes is ongoing. Another challenge is omics studies. Like you understand these metabolomics, genomics, etc. They pose another challenge because they need a lot of data, quality data that needs to be generated, that needs to be documented and made available for retrieval of information. For online structural determination, HPLC MS represents a key strategic tool that can be used mainly for the dereplication purposes. So you don't want to lose your effort doing the isolation or the analysis of known compounds. So thereby you have to develop MS databases to restrict the possibility of using the same process again and again. The use of biological, or what we say as affinity chromatography in drug discovery, is another due for the future. Many developments have been made in this area during recent years, uh, especially for HPLC and TLC based activity profiling of and HPLC based on flow antioxidant assays. So they have developed and they have come up very nicely. Newer strategies or newer stationary phase, newer HPTLC patterns for separation of of natural products are highly desired. So these are some of those challenges and scope of this natural product analysis. So students, let's now summarize what we have learned in this module. Natural products, they include regulated and unregulated organisms or natural products. They also include semisynthetic derivatives, pure compounds which have been modified, from the which have been designed on natural scaffolds, or which have been designed by an inspiration from the natural compounds. Next is that we learned that chromatography and spectroscopy are the conventional methods used in the separation and identification of natural products. The same two methods remain the mainstay of the analysis of these natural products. Hyphenated techniques combine the separation technique like chromatographic methods with the identification methods like spectroscopy. So it takes the advantage of both these methods for the analysis of natural products. And the last of all, progress in natural product analysis is strongly linked to the advancement in the innovation related to the detection and separation methods. Thank you.